Welcome to another video on VAIDR, and this time we want to talk about iPSC cell culture and how to control it, how to make sure that the cells are doing okay, and how to get those quality metrics in an efficient manner without disturbing the cells, without having to stain them, without having to kill them, so you can really focus on their quality and use them for those subsequent experiments that are probably very important to you. So the VAIDR system consists of a label-free automated microscope and a very powerful AI-based analysis software. The software is already pre-trained to do a number of things. In this case, we want to talk about the iPSC analysis that's already built in. So when it comes to iPSC analysis, things really couldn't be any simpler. You take your cell culture vessel out of the incubator and put it into the VAIDR microscope. Then with a push of one button, both the measurement and the subsequent analysis are started. Okay, so let's get into how the analysis works a little bit. So here is an image that was taken by the VAIDR microscope of an iPSC colony, and you nicely see the cells, the borders, the nuclei and so on. And the first thing that the analysis does is to find the nuclei, and the second thing is to find the cell area of the entire colony. That's basically it. Everything else can be derived from this. So at this point, what really comes in handy is the data management capability of the VAIDR system. If you record a single plate repeatedly, let's say over the course of cultivation for your iPSCs, the system will already track that and will know that it was the same plate. So it's very easy then to generate time courses, for example. So you can track the cell growth, or any parameter that's calculated by the iPSC analysis can be plotted versus time. In this case, we have tracked two wells on a 6-well plate over the course of their cultivation. One well was seeded with a higher density and the other one with a lower density. And as you see, these two growth curves are nicely separated and automatically tracked by the VAIDR system. The whole purpose of this is that you can focus on making decisions about your cell culture, so you shouldn't be concerned with the imaging or with the analysis. So let's get into some of the data interpretation that we can do based on these two wells. So we have a confluency curves that basically shows the percentage of area covered by the cells, so basically your cell growth. Also the nuclei are counted, so you can also interpret the cell growth in terms of numbers of cells. You can also divide these two quantities to get the effective area per cell, and this is also quite interesting because what you see here is that it decreases over time. Well, it makes sense, right? The space for the cells to grow into diminishes because at some point the plate starts to be full, but cells still keep dividing, keep growing in numbers, so the area per cell is going down. So up to now I've talked about features that are already included in the iPSC analysis module. What I haven't talked about is a feature of VAIDR that's quite useful if you want to go beyond that. So if you click on some of the other videos on this channel, you will see that VAIDR can be trained to detect structures of interest to you. And I just want to highlight this in a very small example because there's one more feature in the iPSC colonies that might be of interest concerning quality control. I'm talking about the cracks between the cells, so they give an impression of the density and of basically the compactness of the iPSC colonies. And as an example of training VAIDR to detect arbitrary structures, I just want to show you that we can highlight these cracks and of course also quantify the area, the sizes of the individual cracks and so on. So this should give you an impression that VAIDR can be customized to any need that goes beyond the already present functionality. If you're interested to learn more about the VAIDR system or have a question for us, just drop us a line via email or visit our website. One more thing, as you've noted, this channel is still quite young. So if you're interested in upcoming topics on the VAIDR system and how it can help you with your cell culture, just subscribe to the channel and always keep up to date with our newest updates. That's all I got for this video. I wish you a lot of success with your iPSC cultures.